Hello students, so welcome back to the channel. In this video, we are going to study about the seed dormancy. We'll be focusing what are the reasons for the dormancies and what is difference between the seed dormancy and quiescent seed. So see here, first of all, try to understand what is difference between quiescent seed and dormant seed. See, if the seed is not able to germinate, means it's a period of the suspended growth and it is not able to germinate because of some exogenous factor like environment condition. Then we say seed is quiescent seed. So it is a suspended stage of the growth and seed is not able to germinate because of exogenous factor. Okay, like your temperature or your moisture, whatever be the other reasons. The seed is unable to germinate due to exogenous factor. And these exogenous factors are also your environmental factors, okay? Generally, they are your environmental factors. But if suppose the seed is not able to germinate because of some internal factor, then we say dormant seed. So in the dormant seed, we are finding suspended growth and seed is unable to germinate because of some endogenous factor. Due to endogenous factor. So first of all, basic difference is clear. What is difference between the quiescent seed and the dormant seed? Clear? So this is under the control of the exogenous factor and dormant seed is under the control of the endogenous factor. So endogenous factor can be your like metabolic factor only because of some metabolic conditions. Is it clear? So now let us study the reasons for dormancy. Dormancy. First, it may be because of seed coat. Either the seed coat is impermeable to water or it is impermeable to gas. Many reasons can be there due to seed coat. So it may be impermeable to water. Okay. It may be impermeable to gases. Or it may be mechanically, it is so much hard you can't break it. So it is mechanically resistant. So these are the reasons and it's because of the seed coat and the plants in which we find such types. For example, if it is dormancy is because of the seed coat is impermeable to water, then we find such conditions in trigonella. And it is if it is not able to germinate because it's impermeable to gases, then we find such conditions in apple. And here we find such conditions in amaranthus. So because of the seed coat, these are the few reasons of the dormancy. Now it may be that the embryo is not mature. Another reason can be due to immature embryo. So it is taking a time for the maturation due to the presence of immature embryo. And such conditions is seen in one example of the gymnosperms, that is Jingo biloba. So this example is present in the gymnosperms, which is a connecting link between the cycas and the pinus. Then it may be that they require a low temperature treatment due to requirement of low temperature treatment. And such conditions will be seen in one plants like a polygonum. They require some specific light due to specific light requirement. Few seeds they require the presence of light means they are actually positive photoblastic seed. Photoblastic seeds are there. It means they require light requirement. If they respond to the light, we call it as positive photoblastic. And if they are inhibited because of the light, then we say negative photoblastic. The photoblastic seeds are those seeds which show response to the light. If they are germinating in the presence of light, means positive means they require specific light for the germination. Is it clear? Now, example you can understand this way. One plant is there, that is in the lettuce. We are finding red light is promoting germination. In the red light, germination is going on. But in case of the infrared, if you give infrared light, then germination is inhibited. Is it clear? So they require specific light. So we say they show response to the light. That is why we say they are positive photoblastic seed. And few are the one which are negative photoblastic. Negative photoblastic means they'll be inhibited. You can say they will not germinate in the presence of light. They inhibit the germination of their seed. And for this best example we can take is your onion we can take. So I think so till up till here, all these conditions which are necessary reasons for dormancies. I hope up till here all the four types reasons are clear to you. Now let us see another reasons. Sometimes it might be that the inhibitor might be present. So due to the presence of germination inhibitor, which is not allowing them to germinate due to the presence of germination inhibitor. Best example you can find it, one chemical that is ferulic acid in tomato. Ferulic acid is an inhibitor which is present in the tomato pulp. It is an inhibitor actually. It is a response, it is a reason for the dormancy. Seeds are not able to germinate because they require some time, or you can say they want particular hormone. So you can say they are requiring or they are demanding of some after ripening period. Means they will mature embryo is there, they are having mature embryo, but still they want the presence of certain hormones. That is why they are not able to germinate. It still requires the presence of certain hormone. Is it clear? So they want certain time after ripening also, they want certain time so that they can develop that hormone and then they can germinate. Example, best example, you can see all your cereal crops like wheat, your barley and all these are the few examples. So these are the various reasons for the dormancy. Now, how can we break the dormancies? There are various methods how you can break the dormancy. Number one is the stratification. Stratification, and number two is scarification. See, I'll give my learning tips how I've learned. Here T is coming means it require temperature. Here C is coming means it require chemicals. Okay. The stratification means it require low temperature. You have to give low temperature along with oxygen. And scarification means we have to weak the seed coat. This is successfully for seed coat. Weak seed coat, make the seed coat weak by application of certain acids or alcohol, anything by acid or alcohol. Is it clear? Or you do one thing, you give them the specific light requirement, what they want, light exposure. And last method is, this is very important. They asked in the question also. Low temperature you give. Along with low temperature, use gibberellic acid plus oxygen. This is the way how you can break the seed dormancy. So question is asked with respect to which PGR is used to break dormancy. It is gibberellic acid. That is why if you remember, we have already studied कि ABA, abscisic acid, is antagonistic to gibberellic acid because abscisic acid promotes dormancy and gibberellic acid breaks dormancy. If you want, you can revise through the PGR. Check in the playlist plant physiology, that is plant growth and development. In that you can check it. And gibberellic acid is the one which is responsible for the induction of alpha amylase in the barley. So it means generally it is responsible for the germination of the seeds. So I think so this much topic is clear to you. Thanks for watching.